Our speaker today is Dr. Schönberger, and she's a postdoctoral researcher at the Helmholtz Institute in Freiberg for resource technology. And today, it's a great pleasure for me to introduce her and her talk today. Dr. Schönberger was born in 1990 in Dresden and started her academic career in 2010 at the TU Dresden. In 2016, she graduated and started her PhD in the same year at the Helmholtz Institute Freiberg for Resource Technology, where she obtained her PhD in 2020. Quite early during her scientific career, she focused on the development of peptide-based materials for the recovery of critical metals from industrial wastewaters. Additionally, she is a great inspiration on how young scientists can manage today the challenging balancing act between starting a family and launching a postdoc career in science. So maybe you're also allowed to ask questions in this way, because it's a hot topic today in the scientific field. So with this, I would like to finally hand over the word to Dr. Nora Schönberger and her exciting talk. So please enjoy.
So good afternoon, ladies and gentlemen, and thank you, Michael Erkelens, for the very nice introduction. I'm really glad to be here and have the opportunity to talk to you about biomolecular recycling and about the application of Phage Surface Display for the development of tailored peptides for the recovery of gallium and other high-tech metals. So the first question you might be asking yourself is, why are we in need for such a very special technique for the recovery of metals? So during the development of our society, we firstly entered industrial evolution, and during this process, our demand for different metals increased, and also the quantity of different metals that are used in complex or more complex high-tech products. So today, we are consuming a lot of high-tech products in computers, in our smartphones, for renewable energies, in electronic vehicles, and also for very basic LED lights. This increased demand for raw materials and also the complexity of high-tech products stands in sharp contrast to an uneven global supply of raw materials. So, of course, resources in our Earth are not even distributed, but there are special deposits and special suppliers of relevant high-tech metals worldwide. So our lifestyle and our industry, and by this also our economy is highly dependent of this, of the supply chain and of the distribution of high-tech metals, so that we keep developing towards a more networked society and more online society with a lot of high-tech devices. What is the problem about this? So, sorry. Okay. Resources are a global challenge because we are facing nowadays major difficulties in the resource extraction. The metal content in the earth in mines is depleting, so it's decreasing, and available ores have a more complex body. This, combined with the huge environmental problems in the extraction and the mining and processing of metals, leads to an decrease or an more difficult situation in the supply of raw materials. So we have a high demand, but a lower supply of metals. And this is combined with today's still very low recycling rates for most elements. Conventional methods for metallurgy and processing and metal extraction cannot solve these problems. So today it's not possible to recover and recycle metals from end-of-life products in such quantities to meet our need for those high-tech metals. And that's why new technologies are needed. The development and of new technologies and strategies to meet the demand of industry and society have to enhance the recovery and the processing of ores, makes them also environmentally friendly and more sustainable for nature and society, and possibility to meet these needs is, of course, the recycling. So the processing and reuse of already mined and already used raw materials. The advantages compared to mining are on hand. So it goes with a reduced energy consumption associated, of course, with lower carbon dioxide emission and lower water consumption. So the impact on the biosphere is reduced also because we are not changing landscape so much as by mining. And the production of waste is also reduced. So where does such new technologies come from? It comes from where I come from, so from the Helmholtz Institute Freiberg for Resource Technology. It's a national funded research institute located in Freiberg, near Dresden, and it was founded in 2011 and is integrated into the Helmholtz Center Dresden-Rossendorf. It works in close cooperation with the Technical University Berg Academy in Freiberg. And at the moment, we have around about 140 employees and students, and we are very international from about 30 different countries. But this is, of course, changing a lot. So our directors are Dr. Jens Kutzmer and Dr. Simone Ratz. What are we doing at Helmholtz Institute Freiberg for Resource Technology? We try to focus our research towards a circular economy. So what we are doing is we are researching along the value chain. So from the very early exploration of available mines and ore sites worldwide, we are developing new and more effective extraction strategies from for metals from such ores and from more complex ores, but also end-of-life products, for example, like electronic waste. We improve processing processes for the gaining of metals from scrap or from mineral particles, so to have higher yields and a more efficient production. And the working step also include general metallurgy and recycling. And in this Helmholtz Institute Freiberg, we have also the biotechnology department I belong to, and we are settled kind of next to the processing department, where we develop mainly techniques based on biotechnology processes to separate earth or filter and regain metals from solution by using biomolecular technologies. So our focus is the development of biotechnology techniques for the recovery of metals from raw material sources. So from ores, but also from secondary raw material sources for recycling purposes. Currently, we are 8 PhD students, experienced scientists with a PhD before the name, 10 PhD students, 5 engineers, and 3 students, and we are still growing. Our vision is to combine classical research technology with biotechnology toolboxes to achieve a more sustainable metal recovery. What we can provide are surfacing binding, mineral dissolving, transformation, and metal complexation techniques by the application of the natural properties of organisms, like their metabolism, like their special surface properties, by the production of specialized biocomponents or biomolecules. And this allows us the interaction and recovery from minerals, special materials, and from metals and metal ions. The biomolecular toolbox I like to go into detail today is the directed evolution of peptides by Phage Surface Display technology. Bacteriophage is a versatile vehicle for the display of randomized proteins and peptide sequences, and it was first described in 1985 by George P. Smith, and he used it for the analysis of protein-protein interactions and the development of new biological drugs and the search of specific antibodies for therapeutic, diagnostic, and experimental applications. His work was just a start, and in the meantime, very groundbreaking because he created, by using the Phage Surface Display, a really easy vehicle for directed evolution. And for his work, he was in 2018 awarded the Nobel Prize in Chemistry. So how does this work, and why are peptides a suitable ligand for resource recovery? So peptides are basically short amino acid chains. The biology of those biomolecules provides a huge variety of binding motifs because peptide consists of amino acids with different side chain functionalities. And this combines a lot of different structures that are able to interact very, very tightly and specifically with organic, but also with inorganic surfaces and molecules. So we like to picture our peptides or our bacteriophage as a key chain, and only one of those keys, so one special peptide sequence with an individual amino acid sequence and subsequent secondary or tertiary structure, is able to fit the lock. So only one key fits the lock, and only one peptide is best suitable for a certain target and can interact with a surface or a special molecule. So to look at this technique in more detail. So Phage Surface Display technology mimics a natural selection process, and this selection process is called directed evolution. It is mediated by the bacteriophage vehicle, which presents on its surface randomized peptide sequences. The bacteriophage particle is allowed to interact with a target material, for example, mineral surface in our case, or an immobilized metal ion. Weak binding phages are removed by stringent washing conditions, and only tight binding phages remain. So tight binding phage can be removed from the target material by special elution processes, can be in chemical or biological elution. And that's the very trick about Phage Surface Display. The tight binding phage particles that are eluted can be reproduced and amplified in a special host strain. In this case, it's an E. coli strain, which is able to build an F. pili. And it uses the cell own replication mechanism to reproduce the phage particles. And with this enriched bacteriophage library, we can repeat this biopanning process. This is what the Phage Surface Display affinity selection process is called again. After multiple rounds of this biopanning, individual phage clones are selected, or the whole phage pool is analyzed for the enrichment of certain peptide motifs. We use the Phage Surface Display technology for the development of novel biomolecules for the resource recovery. So in detail, in our labs, we have two main focuses: metal ions and dissolved molecules, and solid target materials like raw earth elements, mineral particles, or plastic surfaces. And we use this technique to develop technical applications for remediation, recovery, or recycling processes. So now I want to go into more detail about one example for the application of Phage Surface Display for the development of a gallium-binding peptide. So goal of this project was the recovery of gallium from production waste in the semiconductor industry. So goal was the objective was to avoid the landfill of residual materials and a recovery, of course, the valuable materials like gallium from the waste, and develop a biotechnological process for the selective separation of gallium from arsenic. So those wastes that contain gallium and arsenic in considerable amounts occur during the semiconductor manufacturing process in considerable amounts and in liquid and solid form. The composition of those wastes is very different and the concentrations vary a lot. So it is not able, or we are not able to apply uniform treatment strategies. Especially challenging are solutions with a very low metal concentration, but also a high metal complexity. So there are a lot of different metals included. The pH value of such low metal concentration solutions vary between pH 8 and pH 9, and the metal content is lower than 0.2%. Those special challenging solutions include also other impurities like iron ions, aluminium ion, silicium oxide, fluor, and other organic compounds such as detergents or surfactants. So this was the target material, so a real wastewater from industry that contains gallium and arsenic in low concentration, but in such amounts that recycling is necessary from an economic point of view, but also from an ecological point of view. And what we did is we developed gallium-binding peptides for the recovery of the gallium from the solutions by using the Phage Surface Display technique. In the first step, we created an experimental setup for the biopanning. So we had to, because we wanted to identify metal ion binding peptides, we had to immobilize the metal ions so that they are viable for the affinity selection process. Then we performed the biopanning in multiple rounds, where the bacteriophage library, it was an PhD 12 random peptide library, so 12 amino acid long peptides were presented on the surface of the bacteriophage, and they were allowed to interact with the immobilized metal ions. By washing and amplification and elution of the interacting bacteriophage, we enriched gallium-binding peptides. So, but what one crucial step in this experimental setup is the target immobilization, and that's why I want to go more in detail with this. So metal ions are especially difficult target for phage surface display because in general to allow a selection of bacteriophage, the target material have to be solid or in somehow immobilized on a surface to have the possibility to interact with the bacteriophage and to also select a binding phage. And for metal ions, we use chelating matrix. For example, we used in the past monolithic IMAC materials, for example, for the selection of cobalt, nickel, rhenium, gallium, and arsenic binding peptides. But we also use pure cube NTA agarose for the immobilization of different metals. So we tested in the past a lot of different materials, and it's quite good working for cobalt and nickel and other transition metal ions that are bivalent, and for all trivalent metal ions that we have tested so far. So we tested rare earth elements like neodymium, europium. We tested gallium, indium, and iron, and other high-tech metals. So, and by immobilizing those metals to the NTA agarose, we get a solid target on which surface the bacteriophage are allowed to interact with the metal ion that is chelated by the resin. Okay, performing this experiment, I identified 5 different potential gallium-binding peptides, and I decided to evaluate the binding abilities of those peptides in individual bacteriophage clones experiments. So after the biopanning with immobilized gallium, I choose 5 different individual phage candidates that were enriched to examine them in biosorption experiments. And what I investigated was how those bacteriophage clones behaved during biosorption to immobilized gallium, but also free gallium at different pHs, and also whether they prefer gallium or arsenic, or whether they have a specific or an affinity for arsenic at those pH, because a main goal of this project was to develop a biomaterial, a peptide that was able to distinguish gallium from arsenic in solution, and thus with high affinity and high specificity only recycle one of those metals and not both at the same time. My colleague Sabine Mattes did this experiment in a major elaborate way. So she used this technique and also the NTA agarose for individual phage with individual phage candidate, and used the setup to determine adsorption isotherms and determine by this binding affinities of single bacteriophage clones for the target material, for the immobilized cobalt or nickel in this case. And what she did was really, really huge experiments, a lot of work, and it resulted in very nice data, and she was able to get binding affinities for the bacteriophage clones, and get two different peptide sequences that were able to distinguish between cobalt and nickel to some point. But working with bacteriophage clones is not what we want in the end. So what we want is peptides that work independent from the bacteriophage clone. So this experiment of evaluating the binding affinities or binding characteristic of individual phage clone that present a special peptide sequence are very useful and insightful, but not what we want in the end. And to optimize this process, we are implementing at the moment a bioinformatic routine for the peptide identification and optimization. So instead of verifying individual phage clones, we use next generation sequencing to evaluate a whole phage pool after illusion after multiple biopanning rounds. And by this, see whether special binding motifs or individual peptide sequences are enriched and use directly those peptides for characterization of peptide interaction with the metal without the bacteriophage, and to develop technical applications based on these peptide sequences. So how do we characterize the interaction of metal ions with peptide? One very useful tool is the Isothermal Titration Calorimetry. It allows us to determine the heat change during the interaction of a peptide with the metal ion, and from this experiments result in parameters concerning the binding mechanism of peptide with metal ion. So we get information about the binding enthalpy, the binding affinity, and also about the stoichiometry of this type of reaction. This is what good or not so good experiments, for example, for isothermal titration of gallium-binding peptides with gallium could look like. So on the left-hand side, first and second diagram in this row contain data for gallium-binding peptides under different conditions. So in one case, in presence of an acetate buffer, and in the second figure in absence of a buffer but at a certain pH. And the last two figures are from a peptide that interacted not as much. So this is what the results from such an isothermal titration calorimetry look like. And at the end, when we do this for all relevant peptide clones, we get something like this. So a lot of data that helps us to evaluate the metal ion binding properties of our selected peptides. So we get information about the binding affinity, the stoichiometry of this complex, the binding enthalpy, and other values that help us to evaluate the quality of our binders and how good they are suited for technical applications. I use this data to choose one peptide for to develop an biocomposite material based on one peptide with the most with the most promising combinations, and I gained this data by immobilizing the peptide to earn polystyrene material and making biosorption experiments with real wastewater and model solutions with this metal. And what I got was a biocomposite is what able to specifically bind gallium and also release it afterward, what is important, and also prefer the binding of gallium over arsenic. So So, and what we can see in the diagram in this slide is that raw material, those peptides, those peptide ligands that showed really all five showed from the beginning really promising gallium-binding interactions. In the end, in a real technical application setup, show very different results towards a gallium specifically. So they have different properties and are differently good or worse suited for the application. And I decided that the C315 peptide was most suited for an application because it shows the best specificity of gallium over arsenic and the best release of the metal afterwards. So the recovery rate was for this peptide best, but not the but it was not the best peptide binder that I gained. So this was really insightful. What else is to consider when working with peptides? Peptides are very expensive, and they are by producing peptide in a chemical way, what is the most used approach to get a peptide nowadays, a lot of toxic waste is accumulated. So we hope that we can transform or develop an efficient peptide expression and purification system based on bifunctional peptides by the application of recombinant DNA techniques. So we want to produce those peptides not chemically but biological by a host organism, and by this, we hope to get a real peptide-based technical application that is also from an economical point of view feasible. So, to conclude, Phage Surface Display technology allows us the development of specialized peptides by the complementary assembly of molecular architecture. So keyword is directed evolution. And these peptides can selectively bind metal-containing particles of metal ions, and the immobilization of peptides on support materials result in stable biocomposites with high affinity for specific target materials. So we get a technical application material. Those peptide-based biocomposites can be used in innovative recycling and separation processes, and peptides are expensive, but recombinant production might in future allow application at a technical scale. Thank you very much. This was my presentation. I want to remind you that the future is circular, so let's focus on recycling and creating a circular economy, and all your questions are welcome. Thank you very much. Thank you very much.
So thanks a lot for this quite nice talk, Nora. So it was really impressive, and also your work. So it was our paper of the month recently. So it's a really nice work. So congratulations, and thanks again.
Yeah, thank you for the opportunity to give this talk, and also for awarding me paper of the month. I was really honored and happy about it. Yeah,
oh, you're welcome.
Also, thank you for all the very, very nice remarks in the chat. I feel like I got personal cheerleaders during this presentation. So this is really nice. Thank you.
Yeah, I think it's a really hot topic, right? So the recycling and resource management is really important today, and so it's a really important work, right? So maybe I'm allowed to directly ask the first question. So then we will change to the live Q&A session. So are you currently, let's say, designing or engineering a kind of prototype filters, or to test this with real wastewater, or is it at the moment really at the lab scale?
It's totally in the lab scale. So I concluded these works, in like 2019 when I finished my PhD work, and ever since I'm really involved in getting enough peptide from microbes or recombinant peptides for a technical application. So our goal was to let this rest until we have a solution for this kind of.
So it's at the moment more a peptide question to bring this in the industrial scale, right?
Yeah, it really is, because peptides are so expensive that such a material would be not economically to use just to recover gallium. Gallium is valuable, but not valuable enough for such an application.
So maybe you can change to gold, right?
Actually, gold is not that expensive, concerned with other techniques, or not as important, right, for the industry. But we are really concerned with getting enough peptides, and we are close to develop strategies or better routines for providing enough peptides. And we also are in cooperation with a startup next to you. It's called Nora, and it produces recombinant peptides. And we had an transfer project with the startup together where we tested one of these peptides.
Nice.
It was also really interesting.
Yeah, this sounds really promising. So we will read a paper, or you will publish maybe a paper in the next
hopefully
time.
soon.
Nice. Okay, so we will have a look. So we're interested. Right. So then let's change to the question and answer mode. So we have a lot of questions from the community. Right.
Okay.
Great. So the first question is from Bernd Lünemann, and he's asking, what is the greatest depletion volume you have already tested? Did you stay in the milliliter scale, or were you testing in the liter volumes?
It's milliliters. So I think during my very early filter experiment, it was like a maximum of 25 milliliters that I tested. And what I also did is testing repetitive rounds of loading and deloading my peptide columns for the recovery of gallium. So, but this was all very small lab scale. So these residual waters or wastewaters are available in large quantities, but my material is not ready for such an application yet.
Okay, nice. So then let's go to the next question from Hugo Diaz. And Mr. Diaz is asking, is this also applicable for other heavy metal ions?
We hope so. So we are constantly developing peptides that are very specific for one certain metal. So gallium was only one project. We also developed up to this point peptides for cobalt and nickel, as I mentioned, the work of Sabine Mattes. We have currently an ongoing project for lanthanides and rare earth elements. We had a look for indium, germanium, and iron and copper binding peptides. So we think this is also applicable, but on the other hand, the development of the peptides is really time and money consuming. So you have for every metal you have to redo the broader process of the screening and optimization and characterization. So yes.
Great. So the next question is from Robert Die, and he would only
It's not a question, but thank you very much.
and he finds your work quite impressive. So then let's come to the real next question. Right, this. So there's a question from Robert D. So he is interested in knowing what conclusions were drawn from this project and whether some of the findings are already being applied.
No, not really. So we are still developing together with the project partners biomolecule-based filters, but not peptides, but siderophores. So a colleague of mine is really into this this special wastewater as a target where gallium is included, because he found a special biomolecules, so siderophores, that it can really selectively bind gallium. And he is involved in a huge upscaling process. So he is the one turning the liters in a higher scale for gallium. So that's also a reason why I've not driven this further. I think in future I will concentrate on other metals when I want to develop a peptide-based application. Of course, the special siderophore Rohan, so my colleague Rohan Jain identified, is so specific and well suited for the special metal gallium.
Thank you. Then the next question was asked by Carmen Silva, and she's asking how far the technology is away from upscaling.
Kind of same answer. So we are so basically we know how to do it, but of course for a real upscaling and a technical application, it has to become economical feasible. So not only the peptide has to be provided in a not too expensive way, but also the material has kind of be optimized, so it have to be very durable for multiple rounds of using. It have to be stable enough also under varying pH conditions and in the presence of different detergents, because the wastewater streams also vary a lot. It's not always the same wastewater, but the concentrations change, the pH changes, the products in it change. So yeah, the process has to be really optimized. So we are we know what we have to do, but we need time and money to do it.
Okay, thank you. So the next question is asked by Thomas Perkins. So he asks, if the plan is to help and supply Biotech with recycled metal ions for IMEC materials.
This is a lovely idea.
Yeah,
never thought about it.
Yeah. So I'm not as I know. So, but it's a nice idea.
I also know nothing about it, but I think this is also kind of a circular lab economy.
Yeah. You're right. So, Robert G has another question. So he's asking what specific hurdles are currently preventing the cost-effective production of small peptides. So if there are some improvements.
So I think I can. I don't want to go into too much detail, because this is really a complex topic. It kind of took my attention for the past 3 years. So to make it very simple, peptides are really small, and to make a good production routine, we have to make them somehow bigger. But when we make them bigger, we also attach kind of a fusion protein that makes the peptide more unspecific. So biomass itself loves to interact with heavy metals, and we want the peptide to be as specific as possible, so that we have this selectivity advantage of the material. And when we try to only produce peptides, the quantities are really small. So, and also, to make it cost effective, the steps of creating the filter material or producing it afterwards have to be much cheaper, because the conjugation of the peptide is it's chemically done at the moment, or at this point when I presented it. And I follow some strategy to make this also more cost effective, so that we have the opportunity to directly link the peptide, also site selective, to carrier material and creative filters. But I hope I can publish the results very soon.
Okay, so maybe we have time for 2 further questions. So the other questions will be answered by mail, so you will get your answer, but sure, maybe later. So Divashre Munishwa is asking, so what the startup was called you mentioned before. So maybe you can say it again.
I think you mean with the siderophores, right? Because it's not with peptides, it's with siderophores, so totally different biomolecule. And the technique, the startup is called Gallium-4 technique. So like gallium and 4, like the metal 4, in one word. And the person to ask all your questions about is Rohan Chain. And if you find me on the HZTR website, you will also find him as a team member. So also, if you write me an email, I can send you the contact if you're interested.
Oh, nice. So in the name of Divrashe, thank you. Great. So last question from, or more or less last statement from Thomas Perkins is Ecospheres. Maybe you've heard about the company, and they seem to do some different, more or less the same work or similar work. Do you heard about this company?
No, I didn't. It's new to me, but thank you for the remarks. This sounds really interesting, so I will have a look at it. Thank you.
Yeah, maybe it's an impression for you that you can maybe get to the application with your techniques more faster, right?
Yeah. Sounds really, really great opportunity.
Yeah, great. So then let's end the question and answer sessions. And as I said before, you will get your answer. So by mail, if there are some more questions, you can also write at contact at biocube.com, cubebiotech.com, and also you can also ask Dr. Schönberger for an answer by mail. Right. So with this, I would like to close this session. But before this, I have an announcement. There will be the next masterclass at the 13th of July. So at the same time, and it's a talk by Toman Bortichen, and he will talk about a semi-automated workflow for the quantitative analysis of the newly synthesized proteome. So a different topic, but also interesting. And so thanks again for joining this session today, and we hope that we will see you again next time. So stay healthy and see you next time. Bye.